welcome. Today we'll be finishing up our discussion of protein synthesis. If you remember last time we talked about a process called translation, where a section of the DNA is copied. That section is called a gene, and that copy of the DNA, which is able to leave the nucleus, is called the mRNA. And so that's basically where we pick up. I want to start with a couple confusing genetic terms and words that people get mixed up all of the time. And so an amino acid is the smallest structural unit of a protein polypeptide. We should kind of remember that from first semester. A polypeptide then is a chain of amino acids strung together. So amino acids, little molecules, polypeptide, a bunch of amino acids strung together to make a bigger molecule. And then a protein is a complex structure, often composed of multiple polypeptides folded and shaped into a specific structure. And then a gene is a distinct unit of material, DNA, found on a chromosome, right? So it's a distinct little segment of that chromosome. So protein synthesis, again, occurs in two steps. Step 1, transcription. Step 2, translation. A little definition for translation is the mRNA, with the help of a ribosome, forms a chain of amino acids, which eventually forms a protein, based on the information that's contained in the mRNA. So let's see how that happens. So we're picking up mRNA molecule has already been made. Now we need to convert that message on the mRNA into a protein sequence. So the sequence on the mRNA is divided into 3 base sections of mRNA called codons. So basically every 3 letters of the mRNA codes for a potentially different amino acid, right? So every 3 letters of mRNA is called a codon. It looks like this if we're looking specifically at the mRNA molecule here. And the amazing thing is, scientists were able to decode what each combination of mRNA nucleotides, what amino acid they actually end up coding for. And so for this one, here is a codon AUG, here's another codon AAC, and here's a codon UCU. And what we see is that we can use this codon table to figure out what possible amino acids can be made. And so let's just look at this example here. All right, in this example here, I have the first codon as AUG. And so if I look at this table, if I'm looking at this table, the biggest letter starts here in the middle, or the first letter is the biggest one in the middle. So our first letter was A. And then I go to my second letter, which was U. So I look for the U in this next segment here. U is right over here. And then my final letter was G. So I go to G, and that codes for the amino acid methionine. See how that works? A, U, G, methionine. So that means the amino acid that's coded for from the codon AUG is methionine. And then each one, whatever combination I take, can be a potentially different amino acid. So here's another version of a codon table. You might get on your test either one of those, so it's important to be able to read either one of these. Right? So if I'm looking at this one, it says first letter, second letter, third letter. Let's take our example again with AUG. And so I've got A here as my first letter. So here's A. So I know my amino acid is somewhere in this little section. And then I look second letter here. Second letter is U. So A and U meet up right here. And then I just look for the codon. AUU, AUC, AUA, AUG. This one I want. It codes for MET, which is the shorthand for methionine. There are specialized codons that are known as the start and stop codons. AUG is known as the start codon. It codes for methionine. All proteins start with the amino acid methionine. There are also 3 stop codons. You'll see them here. Here's UAA, UAG, and UGA. UAA, UAG, UGA. Those are the 3 stop codons. And a stop codon basically means that the polypeptide gets cut off at that point. And so we picked our first amino acid. So let's look at the codon table, try a second one. So CGU. Here's my codon table. Here's a C. Here's the G. So CGU codes for ARG, which is arginine, which we see right here. And then we can keep going, right? So ACU would code for threonine, GAU, CGU, UCA, GAU, and then our final one we know is for the stop codon. And so that's how it works, right? We have this mRNA sequence. It gets split up into 3-letter codes called codons, and then you use a codon table like we see when we go back here, like this, to figure out which one of those codons leads to which amino acid. There's a couple other complementary tools that are used in this process, so let's talk about them. The first one is a specific type of RNA called tRNA. tRNA, also known as transfer RNA. Its whole job is to transfer amino acids to the ribosomes for protein synthesis. And so this is how it works. On each tRNA molecule, there is a site for the amino acid to attach to, and then it has this other site on it called an anticodon. What happens is if the anticodon matches up with the codon that's red, the tRNA will deposit its amino acid into this ribosome so that it can keep growing the polypeptide chain. So let's look here, right? So this next sequence that's left right here in the mRNA is AAA. The anticodon for AAA on RNA would be UUU. And so if these two things match up, the tRNA knows that this is the correct spot to deposit its amino acid. It will deposit its amino acid in the ribosome. The ribosome will help to string together these amino acids we see here, and then eventually it'll get ejected, go grab another one, and come back. And this is how it looks, right? So the amino acid plugs in. We can see the growing chain here. These will get strung together, and then the next tRNA molecule will come try to plug in and see if it fits. And we see here in my final picture what the completed polypeptide chain looks like with all these strings of amino acids strung together. And you can also see in this picture here that the tRNA molecule, once it gets ejected, it loses its amino acid. Then it has to go back into the cytoplasm, grab another one, and it can be recycled and reused. UGA right here, that means stop again. Stop means this is where the polypeptide gets cut off. And so in this process of translation, we see all 3 major forms of RNA being used. The mRNA is what carries the instructions to make the polypeptide. The tRNA transfers and carries the amino acids. And then the rRNA forms part of the ribosome, which makes the polypeptide. Right? So all 3 of these things work together in harmony to make the polypeptide. And that's the basis of molecular genetics. It's called the central dogma because all of biology relies on this. Anytime anything is made in the body, this is the process, right? A section of the DNA is read off and a copy is made. That's transcription. The mRNA copy then leaves the nucleus, goes into the cytoplasm, and attaches to a ribosome. In that ribosome, there's rRNA, and tRNA is used as well to make this polypeptide chain. And then that polypeptide chain can be combined with other things along with other polypeptides and folded into specific shapes and turned into proteins, which we already know proteins have a plethora of jobs that they do in the body, right? And so this is the process which basically your body is doing continuously throughout your life without you even knowing. And this process is also called gene expression because we have a certain segment of the DNA which is read off That's the gene. Copy of that section is made, and then the gene is expressed and the polypeptide is made. So hopefully that made sense to you. Thank you for watching.